Good afternoon to everyone. So it's 5 o'clock, so it's time for the webinar to start. So first of all, welcome to everybody to this new webinar related to bispecific compound. Today, I'm Florence Fompachassi, and I'm the head of marketing and communication at Oncodesign, and I will be the moderator for today's session. So a few indications before starting the webinar. Jean-François Mirgelet will run the webinar. Throughout the webinar, there will be a series of polls just to understand better your needs and also your needs and your project. So it will be just a few polls. Don't hesitate to ask your question throughout the webinar on the chat box on the right-hand side of the window, and there will be a Q&A session at the end of this webinar, and Jean-François will be able to answer directly to your questions. So before starting, let me introduce Jean-François Mirgelet. So Jean-François has been working for Oncodesign for 17 years. Jean-François obtained his PhD from the University of Nancy in cellular pharmacology of anti-cancer drugs, followed by a postdoctoral position at IRIBHM Brussels in chemokines and chemokine receptors implication in cancer cell migration. At Oncodesign, Jean-François started as a study director and then took the position of technology director, directly supporting our business development activities and working closely with our client to select the most appropriate study designs for the evaluation of their therapies through preclinical studies. Jean-François has been the head of the in vitro sciences department since 2018. In this capacity, he managed both services and R&D related in vitro activities. So before starting, I just have a first poll. So what do you expect by attending this live webinar? To obtain solid information from an expert, to have a general overview of the topic, or to better know about Oncodesign expertise? So you have a couple of seconds to give your answer. So the poll is open, so go ahead. A few more seconds to go, so don't hesitate. I mean, is it just a quick answer? Okay, so I'm closing the poll. Thank you very much. And Jean-François, now this is your turn. So please let us know all about BISPECIFIC evaluation platform.
Okay, thank you, Florence, for the introduction. So I will talk for sure today about bispecific evaluation, and I will start by the various topics that will be discussed during this session. So first of all, a little bit of introduction regarding the various technology available for bispecific structure. A little bit more focus on the one that have the ability to recruit immune cells, and at the end of the introduction, an overview of the clinical situation of those bispecific antibodies. So we have, in fact, two main strategies to define and make bispecific antibodies. The first one is really to address and target tumor cells with various options. So it's only targeting tumor cell by itself, but having multiple components on receptor, on ligand that could block most of the time the proliferation or induce death on the tumor cell itself. On the other hand, we have the opportunity to retarget some part of our body, and more precisely immune cells, to fight against tumor cells. And in that sense, it's really BISPECIFIC, having an arm against tumor cells, but a recruiting function for recruitment and activation of T cells most of the time, and we'll discuss that later. We can also observe some strategy to deliver some killing strategy toward tumor cells, which could be a cytokine or enzyme that will be able to activate prodrug, as an example. So mostly two different strategies, one for the tumor cells itself and only directly against tumor cells, and the other one that really act as a recruiting component for the immune cells. Then, having those two strategies in mind, there is a multitude of formats available for developing multi-specific antibody. It could be bispecific, and we also have now tri-specific structure, and those formats could be as small as SCFV couple structure. So these are the smallest structure having the same properties and an antibody to bind to the antigen, and the main differences will be the PK function, meaning this BITE structure behave quite as similar as a small molecule as compared to a full structure antibody, and for sure a bispecific antibody where you can have sometimes two complete IgG combined together. Talking about format, there is now a lot of questions regarding which format develop the best potency, and we have the chance now to have in clinic and in preclinical situation multiple format for the same target. So the main example is CD19 on the tumor cells and the CD3 arm for the recruitment of T cells, and it has been specifically shown that most of the time the smaller structure have better potency. Like we can observe on this left-hand side part of the slide that for CD3, CD19, the smaller structure is about 10-fold more potent than a full IgG, and against when we simplify the structure with a simple SCFV with two specific functions, it could be even better, meaning 1,000-fold better than a full IgG or this combined structure. Now there is available in clinic a lot of additional target, and then we have also the ability to address the correlation between format and potency. And sometimes there is a surprising observation, meaning this big structure is here recruiting CD3 against CD33 seems to be 24 better than the bispecific complete IgG. So it's difficult to generate a rule, and it's something you have to address by developing various formats and then test which one demonstrate the better potency. Having said that, we have two main structures, the one that is fully dedicated to the tumor cells and the one that is recruiting immune cells toward tumor cells, we have to spend a little bit time on which immune cells you want to recruit and by which way, meaning which antigen, which receptor could be used to recruit immune cells on the tumor bed. First of all, the first one that is fully addressed and then demonstrated to be functional in clinic is the one that target T cells, and two main strategies has been tested, the one against CD3, and now we have some tests with new structure that recruit T cells using a soluble TCR. We now have also in clinic few options on NK cells, and one of the best strategy, at least the one that has progressed to clinic, is the one that recruit NK cells through CD16. And we also have multiple options for other kind of immune cells. But depending on the microenvironment, there is some still difficulties to understand which monocyte macrophages are the best one to attract, and will they stay same phenotype when I've been recruited to the tumor bed. Other strategies that are moving rapidly is the one that is specifically recruit gamma delta T cells instead of total T cells. And the new strategy are really to recruit a subtype of T cells and not the whole T cells through CD3 receptor. Having said that, it's a matter of compound and immune cells, and based on those two actors, the mechanism of action could be changed. And then by recruiting macrophages, you have ADCC and ADCP function, and one of the advantages of that is through phagocytosis, those macrophages could then be antigen presenting cells and then help to develop tumor specific T cell response. By having NK cells, again, you will generate direct cytotoxicity. T cells develop also direct cytotoxicity through release of cytotoxic cytokine, and at the end, granulocytes such neutrophils could also be readdressed toward tumor cells and then generate phagocytosis and be presenting cells. Florence, it's up to you to ask a few questions through Paul, please.
Yes, Jean-François. So now we would like to know, if your compound has capabilities to recruit immune cells. So just a yes or no question. So if you want to give us your feedback, thanks a lot. Okay, a few more seconds to go before I'm closing the poll. So off we go. I'm closing the poll. Here it is. Back to the presentation, Jean-François. Thanks a lot.
Thank you. Then we can have a look on what is the clinical situation right now. So only one bispecific compound is approved today, which is called blinatumomab. It's against CD19 and it recruits T cells through CD3. And that's the first proof of concept, and then it's opened the way for multiple competitive structures. So we have now in early clinical phase various formats against B cells antigens such CD19, CD20, and they all recruit T cells using a CD3 antibody. We have also additional target that is used on against hematological malignancies, and now it's not only available for lymphoid and B cell derived lymphoma, but also for myeloid and cells having targets such as CD33 or CD38. And you can see that most of the structure is recruiting T cells through a CD3. There are few companies that develop NK cells recruiting agent, and as mentioned earlier, the two compounds that is progressing in clinic, one in phase 1, another one in phase 2, is recruiting through CD16A antigen, and they are both dedicated to CD30 positive T cells, tumor cells, sorry. This bispecific antibody has been demonstrated to be a useful tool for hematological malignancies, and there's now a lot of development for making them also functional for solid tumor. And then we can also see here that there are some progressing in phase 2, and against most of them are recruiting T cells through CD3 antibody, and you can recognize famous tumor antigens such R2, such PCMA for prostate, such EGFR for both lung and colon. And there is also specific sub-strategy that combine CD3 and soluble TCR to access really tumor-specific peptide such GP100 or NY-ESO1. So having given you an overview of the various structure, the various format, and the clinical situation, it's the time for me to introduce a little bit of pharmacology. So I will describe few study cases, and the first I will talk about is the work we've done with blinatumomab, so CD3, CD19, and how we develop assays and model to really address response to this compound. First of all, a few words on blinatumomab itself. So, as I mentioned earlier, it is a CD3, CD19 bispecific structure, is based on the DIABODY approach, and this has been the first in-class BiTE antibody approved. So you have the structure here is really as small as possible, meaning 2 scFv, one against CD3 and one against CD19, and covalently combined together. And it was approved in 2014 for acute lymphoblastic leukemia, so leukemia that are or show positive for CD19, and it has been progressed in subtype of the disease positive for this tumor-specific target. So in vitro, I would say that the system is quite easiest. You just have to put T cells against a tumor positive for CD19, and in between your specific compound, what we have developed is an assay where we can not only monitor tumor cell death, but also monitor T-cell response. And T-cell response could be of two types. It could demonstrate activation marker. T-cells could proliferate during the assays, and then using a flow cytometry-based assay, so we can monitor both cell type, so the tumor cells and the T cells in the same assays format. Here is an example of what we can observe as death. So it's the variability against control, and in red you have the cytotoxicity that is induced by T cells when both arms of the bispecific structure is bound. So one arm to T cells, the other one on tumor cells, and we observe a really good signal. And when we have only the CD3 arm, we can observe some evidence of cytotoxicity, but with really high concentration. So having an arm against tumor cells and creating a synapse between tumor cells and immune cells, you can really see that it improves the cytotoxic effect. Regarding in vivo system, to address efficacy of blinatumomab, you need to create a model where you have human T cells and where you have tumor expressing the right targets. So the model we used was based on NOG mice and used mature PBMCs coming from healthy donor. And the protocol is the one described here. So animals were whole body irradiated in order to improve both tumor cell development and adopted transfer of PBMCs. So the B-cell lymphoma is the Ramos tumor cells, and it has been injected through IV route, and IP cells were injected through IP route, so intraperitoneal injection. In our system, the main readout was based on tumor cells that grow in the bone marrow. And I will show you some results. So mainly we have 3 readouts. So we can analyze survival increase, meaning it's a model where tumor cells development should kill the animals. We can also monitor tumor cells depletion, and this has been obtained using bone marrow puncture. And we also have to demonstrate that immune cells has been moved from the circulation to the bone marrow, where they have to meet the tumor cells. So first of all, we can observe here that blinatumomab increase the survival of the animals. So about 28 days for the median of the control, and an increase up to 35, 36 days. I have to say that we stopped treating early, so it's normal that the animals were not healthy at the end, meaning we know that the treatment we applied is not curative at all. Regarding tumor cell depletion, so we have analyzed the content of B-cell lymphoma in the bone marrow, and we can observe here on those flow cytometry profiles showing that we have here B-cell lymphoma that grow in the bone marrow for the control animals, and in the treated animals we have significantly decrease the number of B-cell lymphoma. And we can also observe that we have an increased negative population, meaning CD19, CD20 negative. So new cells that appears in the bone marrow, and for sure these cells are the T cells we expect to migrate to the bone marrow due to treatment. And that's we can observe using in the graph here, showing that these are B-cell lymphoma. So in the control, most of the cell in the bone marrow of mice is B-cell lymphoma, and we have a significant decrease after treatment. And one of the interesting things is T cells are not going to bone marrow by itself, meaning in the control animals where there is no treatment, a few T cells are moving to the bone marrow, but in presence of the bispecific structure, we have a really significant recruitment, and at the end, we have completely changed the type of immune cells that we have in the bone marrow, replacing tumor cell by T cells. We can ask here the question why PBMCs has been injected through IP and not a more physiological way such IV. And in fact, we have those experiments done comparing the same model, so Ramos B-cell lymphoma injected IV, and then tests how PBMCs when injected IV or IP could interfere with the tumor development. And when I say tumor development is tumor development in the bone marrow, which is the model we know. When you have only PBMC, most of the cells are not circulating in the blood, but they are homing to the bone marrow and then grow in the bone marrow. So on the graphic here, you have on the left-hand side the blood, and on the right-hand side the bone marrow, and the results are PBMCs. So we can see that when you have PBMCs IV, you have increased number of PBMCs in the blood as compared to when you have PBMCs IP. It's totally normal. The kinetic of development is slightly low when you apply PBMCs IP, so it's just a matter of time to have the same number of circulating immune cells. Regarding the bone marrow, in both conditions, you have approximately the same number for the T cells that goes directly to the bone marrow, whatever they are injected IV or IP. Now the question is, what is going wrong or well with Ramos cells? And in the blood, as I said earlier, this is not really the main way for where we can find tumor cells. So we have them, but in a really low number. However, most of them are going to the bone marrow, and you have here in blue the tumor cells when they are alone, so without PBMCs. In red, you have the tumor cell number in the bone marrow when you have PBMCs IV, and in green when you have PBMCs IP. And in fact, when you have PBMCs IV, they probably start to kill tumor cells in the blood before they reach the bone marrow. And then at the end, the route of administration of the PBMC really modify the physiology of our model, and then it's why we have developed this model having PBMCs IP instead of PBMCs IV. Florence, it's up to you for a new question.
Yes, yes, Jean-François. So I've got two questions to go on. So, have you already faced scientific issue in developing orthotopic model or subcutaneous model? So please choose one of the two answers. So there will be the first question, and there will be a second one just right after the one. So are you facing any scientific issue in one of those two models? Please choose the one you are facing models. Okay, so that was the first one for this series, and I've got another one. If you can let us know which model are you currently working on, hematological cancer or solid cancer? Please don't hesitate to let us know which one you are working on. Which models you are currently working on. Okay, so thank you very much for your participation. So you can carry on your presentation, Jean-François. Thanks.
Thank you, Florence. So I have demonstrated here that most of the time you have some difficulty when establishing a new model for bispecific compound that recruit immune cells, where you have to deal with both the immune counterpart and the way that you have to develop your tumor. So I will also exemplify two other situations. So one situation where we have an orthotopic tumor, and we have used here as an example an ovarian tumor that developed ascites in the peritoneal cavity, as in human. It's really a slow-growing tumor, and so we first inject tumor cells through IP, so that they develop and grow within the peritoneal cavity. Then, around 3 weeks after tumor cell injection, we have PBMCs IV this time, and then we can start to treat, aiming to modify T cells from the circulation and move them to the peritoneal cavity to fight the tumor cells. In this model, I will not describe the details for survival, tumor cell depletion, immune cell recruitment. It's quite the same process we have applied here as compared to the Ramos model for blinatumomab questions. We have here an advantages of having a circulating biomarker like in human. So meaning this of course 3 tumor really CA1 to 5 antigen in the blood. So we can monitor tumor development using a simple ELISA that is performed on a weekly basis using a really small blood puncture. And it gives us two new function. We can randomize animals based on the tumor load here in order to really have an homogeneous tumor development at start, and then use that marker to evaluate how this tumor is progressing when under treatment pressure. First of all, we use that to compare the speed of growth, meaning we know that PBMCs could really interfere with tumor development, and that's the evolution we have here on the graphic, showing that on day 20, it's the randomization days, and we can really observe growth of the tumor through the increase in CA125, and there is no main differences and no change in kinetics development when comparing tumor in presence of PBMCs or tumor only. So no major interference between tumor here and T cells by itself. When you apply treatment, we can easily see that during the first week, there is no main differences between meaning these are the yellow dots here, so showing that between day 20 and day 28, there is no main changes in the tumor load. However, day 42, the level of CA125 is decreasing down to 0 in most of the animals, showing that we have a really nice shrinkage of the tumor here, and it's really homogeneous for all the animals, and it demonstrates that the treatment was started day 22, and it takes a bit of time to demonstrate efficacy, and it's probably due to the reason where we need time to recruit enough T cells on the tumor bed. I've presented here two types of orthotopic tumor. One where we have tumor development in the bone marrow, when another one where we have tumor development in the peritoneal cavity. Most of the time, we are requested to develop subcutaneous tumor in order to have an easiest way to monitor tumor development. So again, there are plenty of options, and we have also used those models for bispecific evaluation. And I have here two examples. The one is not really a solid tumor in subcutaneous situation. It's a myeloma. And in this model, there is no changes having PBMCs on board. So having PBMCs IV did not interfere with the growth of this myeloma at the subcutaneous level. And on the treatment, we can really observe that we have two kinds of population. Of one where we have a nice response and a complete shrinkage of the tumor, and another group of animals where we did not observe significant response. And we have two, and it's possible, but we did not do it for this particular experiment. We can try to understand why we have these two profiles, and try to understand why in this population either the T cells are moving and they stay inactive in the tumor bed, or they are not moving to the tumor bed. Another model with a breast tumor as a subcutaneous tumor, showing again that we have good growth for the subcutaneous model, and a really nice response for 100% of the animals. And again, what we can observe here is it takes time to have an answer, meaning here the treatment starts day 28, 29, and we only start to see efficacy after day 42, day 43. So it takes time to have the right number of T cells at the tumor bed playing their cytotoxic role. So you have to develop a model having the right window to treat and the right window to evaluate tumor response. So we know that it's a lot of work to develop those models and having a model for multitude of targets. And we have all seen that in clinical stage, we have already a lot of target that is moving and progressing, whatever it is, solid tumor or blood-derived tumor. And we have the chance to have now available a lot of model, whatever they are, subcutaneous solid tumor. We have also developed various hematological malignancies, IV or as a subcutaneous model. And we also have evaluated some PDX version where we have then the PDX at the subcutaneous level and then PBMCs, IV or IP, depending on the model. And most of the time, we address clearly the question for all the hematological malignancies where we can know we have already observed that PBMC could interfere with tumor development. So for all subcutaneous tumor, we apply systematically the IV route for PBMCs, but in case of hematological malignancy, we most of the time compare for each model IV versus IP in order to be sure that the tumor develop as it should. So as I mentioned, we have now a really large collection of models that is fully validated to evaluate this specific compound. We discussed previously about the ability to have not only T cells recruiting agent, but also new development having subset of T cells. And I will show you some results for specific subset of T cells. So you have on the screen gamma delta T cells, where you have to deal with two types of issue. The first one is to have good numbers, meaning a large amount of specific cells to be able to humanize a large number of animals, perform pharmacological studies. And you have also to be sure that through adaptive transfer, the cells you are, I would say, playing with is able to survive normally in the mouse. And it's not always true. So here we've developed the capacity to differentiate T cells to quite only gamma delta T cells, and we have phenotype them and demonstrate their phenotypic function, meaning the ability of those T cells to develop a really potent interferon gamma signature. And we have also transfer those gamma delta T cells in various mouse strain. And I will show you how they behave in vivo situations. So we have perform IV injection for gamma delta T cells, and then we track them in the blood over the time. So it's the small subpopulation we can find here in the blood of the animals, but it's really a nice teeny population. However, when we try to follow them over the time, we can demonstrate that those T cells are not circulating over more than 10 days. So day 10, it's really hard to find them. And then day 14, they are not in the blood anymore. So maybe two questions. Are they moving elsewhere? So we just don't know, as we have screened a lot of organs and did not find trace of them. And probably they are not able to survive easily in the blood of those mouse. And then you have to set up a model where you have to come back again every 7-10 days with a new batch of gamma delta for having the ability to mobilize them and use them for testing your bispecific compound. Another population that is of interest, and you have seen that there are now at least 2 compounds progressing in clinic that is recruiting NKs. And that's another population which is not so easy to have in our in vivo model. And we have first start with trying to use the NK cells that are part of the PBMCs, meaning in PBMCs you have T cells, B cells, NKs, and monocytes. So we have tracked those NK specifically in the blood of nog animals. And you have here the total leukocytes showing that over the 10 first day, it's hard to track cells, and then it grows rapidly. Within this leukocyte, you have here the specific NKs, and it's really, really low number. And when we make the proportion, we can demonstrate that at the end, it's less than 1%. And in parallel of the growth of T cells, which is the main proliferating population in PBMCs, humanized animals, those NKs are not growing at all, meaning they became the minority, and it's hard to use them to readdress and make them potent enough to fight the tumor. And I guess it's again the time for Florence to come with the new questions.
Yes, this is Paul time that I would like to say. So here it is, the question: what would be your preferred model? Mature PBMC, stem cells, or any other? Once we've gone through what Jean-François just explained to you. So please let us know what would be your preferred model: mature PBMC, stem cell, or other. Okay, I'm just about to close the poll, so if you have not answered yet, please go ahead. Okay, so I'm closing the poll. So back to you, back to the webinar.
Yes, thank you.
Thanks.
And so the main question here is, do we have other option to have good amount of NK in a mouse system? So we can play and analyze those subpopulation in stem cell based model, and as demonstrated by us and now nicely described in many papers on regular stem cell based model, we do not have the chance to have a nice reconstitution for NKs, and when we have them, they are functionally arrested, and the main reason for that is they are missing cytokine to survive and to be fully functional. The two cytokines that help NK to grow is IL-2, IL-15, and you can then test transgenic animals that are humanized for those two cytokines, or transgenic expression of IL-2, IL-15, and by chance they are developing a really nice NK population, fully functional. We've demonstrated that in vitro. The main drawback in the model is at the end, and you can see that here, we have only NKs. So that's not what we want to have, meaning it's not representative of the human blood. However, it's a compromise, meaning there are various options, but you have to deal with number of or a functional level for this particular NK model in humanized mouse model. To close the presentation and the scientific part, I just want to mention that you may want to see your immune cells moving in the body of the animals, and at Oncodesign we've developed an in vivo pharmacology platform, and we have already the expertise to make your bispecific structure available and compatible with imaging, and for that most of the time we are using bioconjugation and radio labeling, and it's a bit of chemistry with a chelating agent, and then the selection of the appropriated radioisotope. You have then to confirm that your compound is still fully functional, having been modified with the chelating agent and having also the radioactivity on board, and then move that to in vivo situation, and then using SPET or PET machine to track your immune cells moving from the circulation to the tumor in the presence of the bispecific compound. Then, as a conclusion, I just want to mention that at Oncodesign we have the complete expertise to progress your compound from the drug discovery stage to the early clinical situation, and I've developed a lot of preclinical tools to really understand the power of your compound. And recently we launched a premium solution that has really extending capabilities, helping customer to progress their compound from hit finding to IND. It is more specific for small compound, small molecules, but through the platform you have a lot of capabilities that could address bispecific characterization through biophysic analysis, and for sure pharmacology, the main topic of today. And we can also help customer to move their compound to preclinical regulatory stage up to IND. And I would like to thank you for your attention, and I'm happy to take a few questions.
Okay, so thank you very much, Jean-François, for your very consistent and solid presentation. Before just going to the Q&A session, just one more question, the last one. I mean, just to know exactly what would be your the top criteria to work with us. Is it because we are an innovative? We are innovative and propose fit to purpose approach, because we have a one-stop shop expertise, or because we have seasoned project manager to follow up your project. So please let us know a few seconds, and then we can go to the Q&A session. A few more seconds to go, and we will be closing the poll before going to the question, because I can see that there is quite some coming up. So I'm closing the poll, and now I've got some questions coming up. So Jean-François, the first one is my compound structure as a functional FC. Do you have an assay to address FC mediated function?
Yes, thank you, Florence, for the question. So there are multiple ways to address FC function in vitro or in vivo. In vitro, I would say it's quite easy. So you can work on two sides, meaning the complement side and then the immune recruitment function, and you can play with NK for cytotoxicity or neutrophil for phagocytosis. That's really easy assay in vitro. In vivo, it's a little bit difficult. You have to have the right control and the good tool to demonstrate the recruiting function through NK through FC, by itself. And most of the time you have to compare an MC available and a killed FC function in order to be sure that you can demonstrate the added value at the in vivo level. At most of the time, it's really discrete added function, and then it changes only the result, but only 10–15%. So it's most of the time slightly difficult to make that statistically significant, but it could be demonstrated having the right in vivo design.
Okay, so thank you very much for this answer. I've got another one coming up. Did you observe GVHD, in PBMC humanized mice, and how do you deal with this?
Yeah, so it's a really good question. So having fully mature immune cells and most of the time T cells, you have to play, if I can say that, with GVHD, meaning it will come at some points. And then, as I mentioned during the talk, you have to adjust and find the right window to be able to evidence the efficacy of your compound. So it's a matter of number of T cells and also a matter of protocol and finding the right number, the right time to add them on board. You can evidence efficacy of your compound before GVHD start to kill your study. So it's something you have to deal with, and I would say it's a matter of expertise to find the right setup set up a model where you have the right window to see efficacy of your compound.
Okay, so thank you again for this very detailed answer. So I've got another one. Biochemistry and physiochemical characterization of bispecific antibodies were not addressed. Did you have relevant essays to analyze bispecific compounds?
Yes, for sure. As I mentioned, we have a fully integrated platform. So for sure, it's include essay to understand affinity of those bispecific compounds through SPR techniques. They have also a lot of additional essay to understand PK-PD relationship. We just mentioned that at really start of the webinar, and we know that having a full IgG structure, it will be eliminated from the mouse within 10, 15 days. So in order to reach the plateau, you have to inject 1 or twice weekly for a small SCFV structure. Most of the time you have to inject once a day or as a full and continuous infusion. So that's also things we are able to understand in order to be able to demonstrate the efficacy in the right condition.
Okay, thank you very much again. So far I don't have any more question coming up. Just maybe before closing the session, just a quick recall to all the remaining participants that you will be receiving tomorrow an email with the link to stream this very interesting webinar from Jean-François. And if you want to also go to our dedicated channel of all the webinars, I mean, you have quite a lot of a collection of our webinar mentioning, I mean, all our expertise on in vivo, in vitro analysis from its identification to IND and so on. Do you have any more information to let us know, Jean-François, or I think that we can close the webinar?
No, and you have the main address of Oncodesign at the end of the presentation, and I will be happy to answer any question afterward, I would say.
Okay, so thank you again, Jean-François. Thank you very much for all the participants being on board for this session, and I wish you a very good day, and take care and stay healthy. Thank you very much. Bye-bye.
Bye-bye.